Assalamualaikum warahmatullahi wabarakatuh and selamat sejahtera. This is chapter 7 mutation. So under chapter 7 we have three subtopics: 7.1 mutation, classification and types, 7.2 gene mutation, 7.3 chromosomal mutation. Hi, my name is Muhammad Nur Azmi. I am your biology lecturer. For these chapters we have 7 marks for UPS 3 and 10 marks for PSPM. So let's start with 7.1 mutation, classification and types. So by the end of this lesson, you should be able to firstly define mutation, classify mutation into gene point mutation, chromosomal mutation, and then state two types of mutation: spontaneous mutation or induced mutation, as for example, exposure to mutagen. Next, you should be able to define mutagen, state the types of mutagen, for example, physical mutagen, for example, UV radiations or gamma radiations, and chemical mutagen, for example, colchicine and yttrium bromide. First, the definition of mutation. It is a spontaneous changes in the amount or arrangement or structure of the genes or DNA of an organism. So mutation will then leads to the changes in amino acid sequence. So this will lead in the change in genotype and phenotype. Since the changes are occurred in the DNA, it can be inherited or passed to the next generation. So these changes in amino acids, in genotype and phenotype, this will later on create a genetic diversity. These are the class of mutation. We have gene or point mutation and chromosomal mutation. Under the gene mutation, we have base substitution, base insertion, base deletion, base inversion. Whereas under chromosomal mutation, we have chromosomal aberrations and alteration of chromosomal number. Under the chromosomal aberration, which is the changes in the structure, we have the translocation, deletion, inversion and duplication. Whereas in alteration of chromosomal number, we have aneuploidy and euploidy, where there are two types of polyploidy, which is autopolyploidy and allopolyploidy. These are the types of mutations: spontaneous mutation, which occur naturally, that change the DNA structure, that occurs randomly and is quite rare. Whereas we have induced mutation, which caused by mutagens, and this will occur at a higher rates. So the types of mutation: spontaneous mutation, these are usually errors that occur during DNA replication, where the nucleotide which pair does not pair up with a complementary basis. So they basically pair up with a non-complementary basis. So these errors, which are then not corrected by the DNA repair system. Another way of a spontaneous mutation to occur is due to non-disjunction during meiosis. For this one to occur, it usually involves the whole chromosome or the whole sets of chromosome to fail to separate. Okay, so usually the homologous chromosome fail to separate in anaphase 1 or sister chromatid fail to separate in anaphase 2. So again, this can affect either one chromosome or it can affect the whole sets of chromosomes. So this is an example of non-disjunction that may occur. Another types of mutations are induced mutation. These are usually caused by mutagens that causes the DNA to be damaged and then alter the genes. So what is a mutagen? This is the definition of a mutagen. It is either a chemical or physical agent that causes mutation, thus increase the rate of mutation. So we have two types of mutagens. We have physical mutagens, for example, the ultraviolet rays, the gamma ray, or the X-ray. And we have a chemical mutagen, which is either colchicine or yttrium bromides. These are the example of a physical mutagen. As we can see over here, this is the X-ray, right? And then another set over here, this is what we use in radiotherapy to treat cancer patient. Okay, So this is usually they will emit a gamma radiation lah to kill the cell, right, for the cancer treatments. Another example of chemical mutagen, either a colchicine, okay, which can be used to induce non-disjunction, or ethidium bromide. 7.2 Gene mutation. By the end of this lesson, you should be able to firstly define gene mutation, state the 4 types of gene mutation, and state the effect of base substitution, which is the missense, no-sense, and silent mutation, and base insertion and base deletion, the effect of frameshift mutation. This is the definition of a gene, or known as point mutation. So it is a change in a single nucleotide pair of a gene, which then lead to a changes in amino acid sequences. So it is usually due to error in base pairing during DNA replication. There are 4 types of gene mutation. Base substitution, where a pair of nucleotide are substituted with a different pair of nucleotide, or base insertion, where a pair of nucleotide is added into the DNA sequence. Base deletion, where a pair of nucleotide is deleted from the DNA sequence, or base inversion, where there are 2 or more nucleotide pairs which are inverted, rotate 180 degrees, and reattach into the DNA sequences. In base substitution, a pair of nucleotide is usually replaced with another pair of nucleotide in the DNA sequence. So what happened over here is that the mRNA codon is changed. The effect of this base substitution, it will create a missense mutation, nonsense mutation, or silent mutation. Another effect of gene mutation is known as the frameshift mutation. This usually occurs whenever the number of nucleotide is inserted or deleted is not in the multiple of 3. So this will cause alteration or changing in the reading frame of the genetic information, where the genes are read in the wrong triplet grouping of nucleotide on the mRNA sequence during translation. So this will result in a different entirely sequence of amino acid from the point of insertion or deletion. I repeat again, starting from the point of insertion and deletion. Here we can see the effect of a frameshift mutation during base insertion, during the insertion of a single nucleotide pair. In the wild type, this is the original DNA sequences, AGA, GTC, TTC, which will be transcribed into a codon as UCU, CAG, and AG, which later to be translated into serine, glycine, and lysine. Right? This is the original sequence. Okay, by adding one nucleotide pair, the reading frame shifted. Okay, so this reading frame over here will be transcribed. Okay, AGA still transcribed into the UCU, but starting from the point of insertion, GGT transcribed into CCA. Originally, it is transcribed as a CAG, but now it is transcribed into CCA, which is coded for the proline amino acids. So starting from the point of insertion over here, we can see that all the amino acids starting from the point of insertion will start to change. What happens if the base insertion insert a 3 nucleotide pairs? So this is the wild type, same thing that occurs just as before, but if we inserted a 3 nucleotide pair, okay, the DNA sequence will be transcribed into a new codon, CCU, but the next DNA sequence still transcribed into the same codon as the wild type. So the GGA transcribed into CCU, which is a codon for a proline, okay, while the CAG is a codon for the glycine, is remain the same as the wild type. So over here, only one amino acid is added. Instead of changing the whole amino acid sequences, it only changed one amino acid. So this is what happened if the insertion only added 3 nucleotide pair. The same thing can also occur in base deletion. 7.3 Chromosomal mutation. By the end of the lesson, you should be able to define chromosomal mutation, state 2 types of chromosomal mutation, changes in chromosomal structure, or also known as the chromosomal aberration, or changes in the chromosomal numbers. State the types of alteration, aneuploidy or euploidy or polyploidy. And state the aneuploidy effect on autosomal chromosome, for example, monosomy 21 and trisomy 21, and the effects on sex chromosome, for example, Klinefelter syndrome and Turner syndrome. The definition of a chromosomal mutation is the changes in either the numbers or the structure of the chromosome. These are the types of chromosomal mutation. First is chromosomal aberration, which is the changes in the structure of that particular chromosome. First is translocation, where a segment of the chromosome is translocated into the same chromosome, only in a different areas, but still the same chromosome, or it is translocated to another non-homologous chromosome. Deletion, where a segment of the chromosome are deleted from the chromosome. Inversion, where a segment of the chromosome breaks off, inverted 180 degrees, and reattached into the same chromosome. Or duplication, where the segment of the chromosome, a particular segment of the chromosome are duplicated into the same chromosome. And different types of chromosomal mutation are known as chromosomal number alteration, which is the changes in the number of the chromosome. So the changes in the number of chromosome can occur either aneuploidy, which involve only individual number of the chromosome, or euploidy or polyploidy, which involve the whole sets of the chromosome. Under euploidy or polyploidy, we have autopolyploidy and allopolyploidy. The autopolyploidy, the changes in the whole sets of chromosome are derived from the same species, whereas allopolyploidy, the changes in the whole sets of chromosome is derived from two different but mostly closely related species. These are the types of alteration of chromosomal number. Aneuploidy and euploidy. Aneuploidy does not involve the whole sets of chromosome. Instead, the changes in the number of chromosome is either by additional or losing one or more individual chromosome, whereas euploidy involve the changes in the whole sets of chromosome. Example are in plants, whereas, for example, we have a triploid where the number of chromosome triple, or tetraploid, the number of the set of the chromosome quadruple, or pentaploid, the number of set of chromosome multiplied by 5 sets. These are the effects of aneuploidy. If it occurs either in autosomal abnormalities, occur in the autosome, or sex chromosomal abnormalities, which occur in the sex chromosome. If it occurs in the autosomal abnormalities, which occur in autosome, examples are monosomy 21 or trisomy 21. But if it occurs in the sex chromosome, the examples are known as a Klinefelter syndrome or Turner syndrome. This is the end of this lecture. Thank you for listening. If you have any question, you may refer to your lecturers, or you may refer to these references in these books. This is all. Thank you very much. Assalamualaikum.